Good morning, 2700 invertebrate enthusiasts. Welcome, welcome, welcome. And start out your Monday with some clouds and some sun, at least if you're here in the greater Guelph area, by congratulating yourself for two rather impressive completions of these word finds. I saw somebody in the last, because I put two up, because you've been so fast, and I saw somebody in the first one saying, had selected doof, and I was like, doof? What is doof? I do not understand. It's food! And yes, congratulations to Lily. That's her first appearance in a while, and we're still waiting to see the Doug dog, but he's large now. It's very hard to manipulate him in front of the camera without causing rampant chaos. Rampant chaos, you say? That's what we're here for. So I will save that. Congratulate yourself and holster your stylo. And we're going to begin. Today it says I'm going to check the chat right away. Sure. There's a nod to Caramel, who's asleep on the bed in the other room. Today we're going to move our way through several different subjects. Some of them are in kind of the chronological order of how we like to unpack things in this course, and some of them are moved from what sequentially would appear later on in the term, and we're going to talk about it today because we have a guest speaker for much of Wednesday's lecture, and I'll get to that by the end. And then there'll be, just as a heads up, there will be kind of a pre-lecture for on Wednesday, maybe in the 5 to 10 minutes. But you'll see that appear on CourseLink, and you can watch it at your leisure. The plans for today is that we're going to talk about bilateral symmetry, and we're going to talk about platyhelminthes, and we're going to talk about entomophagy. And just since you're thinking about food, we can kind of scroll through that one again of the cestode moving at the bottom. This is kind of roughly keeping up, maybe falling behind slightly today and catching up in the pre-lecture with what you're going to be experiencing in the lab this week. So, hello everyone. Admitting all, come on in, come on in, come on in. Welcome to Monday. You've missed nothing except for Lily the dog. Apologies. So we're going to be talking about the bilateria, and we're going to be talking about the platyhelminthes flatworms and several different prominent groups within them, both free-living, marine, terrestrial, aquatic, and parasitic. We're definitely not going to get to the parasites today, but oh boy, they're interesting. Taxonomically, the platyhelminthes sit in Brusca et al. In Chapter 10, but the bilateria is firmly embedded, our introduction to them, these concepts are firmly embedded in Brusca's Chapter 4, where they talk about the architecture of animal body plans. So a reminder that of all of these chapters, graze along in the taxonomic chapters, graze along with me as we kind of pop around this Dunn phylogeny and visit these 30-odd terminal tips that we're going to share and explore and celebrate. But dive deep into Chapter 4 because it will pop up all over the place. Agreed? I can hear all your nodding heads. Okay, here we go. Symmetry. We've talked about symmetry already. On the left-hand side of that screen, you can see, as we've talked about it with that example, with a polypoid form of perhaps an anthozoan, either an anemone or a coral polyp, but one that's exemplified by radial symmetry, so that there's one main axis, the center axis, around which all the various body parts are arranged, radially symmetrical. We challenged the simplification of the world being split into those that are radially, those that lack symmetry, and those that are bilaterally symmetrical with our understanding of the ctenophores. Perhaps this long-lost earliest branching animal lineage containing many convergently evolved traits, because they're characterized by this biradial symmetry. So a radial symmetry that has been compressed, almost instead of an O, it's an oval. So two planes that can separate into perfectly similar halves. And today we're going to be talking about bilateral. No, today we're going to be jumping around the slides as I try and let people in. Hello. So the fish is us. I think that's a value statement because if you make big phylogenetic groups, I think we're all supposed to be fish. Perhaps you talked about that in Vert last term. But this is where there, excuse me, there are two axes passing from one passing from anterior to posterior, and that mid-sagittal plane separates left from right, and then another slice that can separate the ventral from the dorsal. So this polarized on two perpendicular axes. So if we're looking at this sagittal plane, you can see that's the cross-section at the top, and then, sorry, that's the diagram at the top. The cross-section is this slice here at the bottom. So the last slide was from your Brusca text. This image is from the older text, just to give you two different kind of ways to imagine what probably is intuitively familiar to you, but we're going to be talking about specific advantages that come, and maybe costs, but different advantages certainly that come with when within the evolution of metazoans, the solution of bilateral symmetry arose. What were we more capable of, and what pressures perhaps selected for this symmetry out of a world that at the time was perhaps biradially symmetrical, but mostly radially symmetrical? What advantages might there be to being split and divided in this particular way? And welcome again. So one of the hypotheses that has been important through time, and again, this particular one is from Rupert, Fox, and Barnes, has to do with the importance of gradients and how animals respond to gradients, particularly for two needs. One of them is feeding, and the other is for respiration. Now, if you are a marine animal that exists in a diffuse environment where food kind of floats around you, something that is radially symmetrical, like this polypoid form that you're seeing here, that is not needing or not having any polarization in response to the environment. It's not facing anything in particular. In fact, it's facing all things at one time or another. This is an evident solution to an unpolarized environment. Now, along the evolution of metazoans, one of the things that we've seen, and we were introduced to it even within the Cnidaria, and we talked about the adaptations for non-anthozoans, so not the corals, not the anemone, but all of the other cnidarian lineages that have a medusoid form, where they're freed from being on the substrate or restricted to the substrate to move within the water column. That gives a gradient already, a gradient of up and down. So it's still a pretty simple environment though, because there's no real, it's up and down. There is a gradient, or deeper and more shallow, maybe is a better way to phrase that. But we still haven't introduced the idea of a particular gradient that affects feeding or respiration. So let's talk about that. If you're bilaterally symmetrical, which this little plug here on the right-hand side is to represent, that means you're polarized on an anterior, posterior, so you have a front and a back, and you have a dorsal and a ventral side. So the anterior-posterior suggests, as soon as we see this symmetry, this suggests movement. And what we see coincident with bilateral symmetry is nearly across all of the lineages contained within the bilateria is coincident with bilateral symmetry is a concentration of cephalization, or a concentration of nervous endings and nervous systems and the anterior end. And so more opportunity for job division, but certainly mobility and potentially predatory and predator and prey. These ecological relationships come into the retention of this novel evolutionary trait. Dorsal and ventral then suggest a differential response to the environment, in how that perhaps there's an exposed surface dorsally and a feeding surface ventrally at the minimum, or a locomotory surface and a defense surface on the ventral and the dorsal side respectively. So when we're talking about symmetry, the polarization in response to an environment it's an important thing to consider when we think about the evolution of biradial symmetry. Now, in that diffuse environment, There are, of course, gradients. One of them is light, that maybe is implicit to that shallow and deeper scenario that I gave in the previous slide. But I think you can see in this sped up image here of this essentially this cnidarian, this radially symmetrical organism, you can see them moving in response to a gradient. And what I'd like to do is to do some discussions. First thing that I want to do is we've got two pictures here, A and B, and I would like your opinion. Come on. I would like your opinion about which is radially symmetrical, so one axis around which the body can be divided evenly in any particular orientation, or one axis, or bilaterally symmetrical with the sagittal plane left and right and the dorsal and ventral slice. So here we go. Here's your poll. You've got four options. A and B are both radially. A and B are both bilaterally. A is radially and B is bilaterally. B is radially and A is bilaterally. I'll give you a couple of seconds. I see just over half of you have answered. I have Huxley Workman in my head, which is probably in a COVID world the best place to have him, because if he was here in the room, that wouldn't be a very good sheltering in place, would it? All right, great job. Now, let me show you what you hypothesized. The majority of you selected that B is radially symmetrical and A is bilaterally symmetrical. And let me just save that screen grab so that I can put it into the PowerPoint later on. Smith trying to juggle all of the things. And you're waiting and you're waiting and you're thinking, what is the answer? Well, in fact, close that. So, sorry, I thought I had press shared results. There you can see the majority of you had thought B, the final answer, that B is radially symmetrical and A is bilaterally symmetrical. Now, in fact, it's the opposite. A is a cnidarian, a radially symmetrical cnidarian, or a cnidaria is characterized by radial symmetry. B is an echinoderm, and it is bilaterally symmetrical. So before we dive into, or as we dive into dealing with this explosion in your brains, before I jump back to the chat, what I'd like to do is to have you talk about why this cnidarian might appear bilaterally symmetrical and why what is within the bilateria might appear radially symmetrical. And so to do that, I've been making the breakout rooms filled with lovely comfy chairs. We'll spend a couple of minutes there, then you're going to go to the Google Doc and share some of your thoughts there. I'll post the Google Doc in the chat that you'll be able to see while you're in the breakout rooms. So it's confusing. Share your confusion with each other and then share what results from that in the Google Doc, and we'll rejoin each other in a second. And go. Hi there, Sherry.
Good morning.
How are you?
I'm not bad. How are you?
Oh, this is 2021. It's like we were,
And it's Monday.
yeah, we were all thinking this was going to be so different from 2020. Oh boy. The walk-in music gives me a tune for the day, or at least for the morning.
Yeah. The invert playlist has gone public.
Oh, super.
I changed it to public because there were other people indicating they wanted to see it.
Of,
So,
of course there were.
absolutely.
I was thinking about how the next one might be made public with a like a YouTube playlist, but I wasn't certain that you can do a YouTube playlist that people can add to,
I think you can, except that we've asked them for films, so I don't know how many of those would be available.
or even just like previews or in a world, those kinds of where up is down and down is up.
Yeah, it'd be kind of fun to compile them. I can have the volunteers do that. They did the music ones
Okay.
mostly, so they could keep track of the list and we could, do something with it.
I see some people have now jumped into the Google Doc. They are at least there. I don't see any thoughts yet.
I don't have a link to the Google Doc.
I just put it in the chat.
Oh, did you?
Yeah.
I just
Or
haven't scrolled down far enough.
I should have. Yes, I did.
Yeah, there it is.
Good.
Yeah. I keep forgetting to scroll down when I'm.
Cool. Okay, here comes a message. Okay. All right, I see some hypotheses popping up, and so everyone who's attending asynchronously, hello, we miss you, and we hope that you are joining us in a different part of the time-space continuum. And I'll share in a second what people have been adding as they leave the breakout rooms, which are now closed or closing. Let's hop back over to the other part of Zoom land and see. Hello, hello, welcome back. Now, let me jump over to see some of what you guys have been suggesting. So show me. It is the correct doc. There's a different sheet. Somebody had asked in the chat about eating insects. Yes, so always. But there's two sheets. And the second one, or the first one says, why might the cnidaria appear bilaterally symmetrical in the bilaterian array? And here are your thoughts. It may have something to do with the polarity, not the visual shape. That's interesting. We haven't seen the layout of the organ, so even though it may look radial, it is bilateral. Now you're getting there. That's actually quite thoughtful. We may be lacking full view, maybe an adaptation of sorts resulting to the environment, in which case the opposite symmetry is better suited towards its lifestyle. Now, number 4, right in here, give yourself a big pat on the back, because as you'll see in a second, that's some of where we're going. In our viewpoint of the organism, uniform bilateral symmetry on all planes can appear radial. Yes? So there's, and then in the bottom, yes, easier locomotion through the water current. Now, it's not so much perhaps locomotion, but it's a response to what the diffuse environment provides. So let me take that out of the way again. Pat yourself on the back. Let's unpack this a little bit. What we've seen in a number of cases in the evolution of metazoans is the secondary adoption of an alternate form of symmetry. And that secondary adoption always has to do with gradients, potentially with feeding gradients. So in this transition, so the sea pen or the anthozoan or the sea pen that you're seeing here, it is evident bilateral symmetry of a radially symmetrical organism. Sea pens tend to exist nearly exclusively in water with current. And so that orientation it's no longer advantageous to be just a simple polyp like an anthozoan or an anemone, just in responding to all things in all directions. But if you're in a current, now imagine I had hair for one, crazy, and imagine there was wind coming from you to me, and if my long hair was blowing in the breeze, the advantageous response or the advantageous orientation to that breeze, if it was bringing me food, was to face into it. And that is in fact what the sea pens are doing. They're taking their long, frilly, curly manes and facing it into the current because of feeding. Now, how to deal with the... Let's go back for a second so I don't complicate things too much. How to deal with the echinoderm, in that case a crinoid, a stalked crinoid that has a long, big, very visually radially symmetrical. That's about the fact that this bilaterally symmetrical organism exists in a diffuse environment, stalked to the ground, just like your archetype of an anthozoan polypoid does as well. And so again, it's secondarily been selected for this apparent radial symmetry where all of these feeding arms and tube arms are orienting in all directions at all times. Now, the appearance of radial symmetry comes after developmental stages early on that have bilateral symmetry. And so these are secondary adoptions of an alternate symmetry that have to do with gradients, particularly feeding gradients or particular orientation, and allowing the creation of essentially perpendicularness to an environmental gradient. So it has to do with feeding environments, and so the significance of being bilaterally symmetrical has to do in the long term about being able to move through patchy environments. Not everything is just diffuse food floating around you as needed by a radially symmetrical organism. That you can swim or move and reproduce and differentiate, both associated with reproduction, but also associated with particular tasks and jobs. So, we have here directionality in this exam. This is the unpacking of the rest of the example for the cnidarian. Oh, let me pause that just for a second. So remember, if asynchronously, I'm going to show this video from Blue Planet. The link is in the chat, and you can go and look at it. It's a link that talks about echinoderms, and if the BBC finds it in our video, it'll tell us it's copyright, so it'll have to be cleaned out. And so you can take a look at it right now. The Great Attenborough is a better day. So you can see secondary radial symmetry adopted in a number of cases within the bilateria, all having to do with food in all directions and no necessary polarization. So that's a little bit of the unpacking as to why we saw what we did. And I want to make sure, I'm going to jump over and vérifier la chat to see if you agree and you're nodding along. And I don't see any questions. So you're all thinking, we know this, perhaps. You're nodding along, and that's great. I love it. So let's keep going. So that's our introduction to potential advantages to being bilaterally symmetrical, that you can deal with patchy resources, that you can differentiate yourself to the environment left and right, but anterior and posterior, which allows specialization associated potentially with feeding and defense. It allows cephalization and the concentration of a brain or a neural network, the concentration of your neural network at an anterior end. There's a whole, and so mobility, patchy resources, and a response, that polarization allowing a response to gradients. Now, as it sits within our done phylogeny, we've been here within the ctenophores, the sponges, the placozoans, the cnidarians. We talked about the xenocoelomorphs and the xenoturbella, the mystery of the, it's still here, purple sock, on Friday. And it's as clean as it was then, which is to say very clean. Now, without talking about super explicitly about the bilateria, we are of course within the bilateria with the xenocoelomorphs, because that was, remember, one of the mysteries. Where did it actually sit within this phylogeny? So now this large blue block at the top, the bilateria, this contains all of the subsequent visitings that we're going to do, all of the subsequent branches within this simplified Dunn phylogeny. Now you remember each one of these nodes represents an area that we have a lot of confidence in. We have varying degrees of confidence or hypotheses about relationships within that. But we want to talk explicitly now about the bilateria group that we're going to be dealing with, because we're going to jump into the protostomia. So this is not our clade, the deuterostomes that contain us and the hemichordates and the cephalochordates and the urochordates. That's going to be much later on, into the 20th lectures. The protostomes is actually where the majority of diversity in life forms and morphological diversity, I would argue, reside. And so that's within the spiralia and the ecdysozoa. So this is the ecdysozoa in green, including arthropods, and the spiralia, including mollusks, brachiopods, platyhelminth worms, rotifers, annelids, bryozoans. This is where we're going to spend a lot of time in the next little while. It's likely where the majority of the focus of the second lecture quiz will sit. So the protostomes, that large clade there, what are some of the apomorphies that characterize this group? Several of them are developmental. We'll talk about that in a second, that the blastopore becomes the mouth. They're characterized by schizocyly. And so this is not just something you would say when you sneeze, but it's when the body cavity is formed by splitting of the mesodermal embryonic tissue. And they are triploblasts, they are triploblasts in the same way that we are, as we talked about with endoderm, ectoderm, and mesoderm. And you can see a figure at the bottom here kind of going through this developmental stages until we have, so 1 to A, B, C, D, across the formation of this body cavity by schizocyly. A super duper word, if you could ever manage it, in Trivial Pursuit, in Scrabble. So on the top, this is a model of the developmental stages of a protostome. On the bottom, which we'll come back to later on, so it's just there for contrast, but I'm not going to break apart those stages. We see spiral cleavage, and then the formation of the blastopore, its eventual, which is that kind of opening that's sealed in. That's going to become the mouth in your worms and in your mollusks. And you can see the coelom, the body cavity, being formed, just as we talked last time, by these derivations of the mesoderm. Now, I give another way of so the protostomes here, this is another just a visual to help you remember. Solid masses of mesoderm split to form the coelom. So we're on the left-hand side now, and the protostomes are, and the deuterostomes are on the right. But you can see the difference, schizocyly, so solid masses of mesoderm splitting to form eventually the coelom, as opposed to enterocoelous organisms, which is what we are, where folds of the archentron kind of vaginate off and pinch off to form the coelom. Now, I will say, developmentally, these stages were really, really important to our understanding of the splits between the deuterostomes and the protostomes. And as with many things that we've understood in invertebrate morphology and evolution for a long time, it's not quite as simple as this. But these are big developmental characterizations of these two groups, the protostomes containing the platyhelminth worms, which we'll talk about in a second. So within the spiralia, there used to be a word, within the protostomes we have the spiralia and the Lophotrochozoa, and spiralia and the ecdysozoa. Now the Tribbiani quotes that you're seeing out there, Lophotrochozoa, just refer to an older word, and I may come back and show you a previous phylogenetic hypothesis about the relationships of different animal groups within this yellow clade here. But the spiralia is where we're going to spend time our next little while. So we have the most diverse groups that we've talked about so far. We'll begin by talking about platyhelminth worms, but this will include our discussions about Nemertean worms and mollusks and annelids. So just between the platyhelminths, the mollusks and the annelids. Those are three of the most diverse groups that we have within animal life. And so there's lots and lots of incredible things to talk about there. And the ecdysozoans will wait till later on in the course. So the Spiralia. Apomorphies for this group include novel methods of excretion. So we see larval protonephridia, which is, so you're looking at a cross-section at the bottom, the donut that you see that has kind of the shape with the box right here at the bottom. Apologies for that jump. This box here is blown up until we see this flame cell. So this is a protonephridia. So this is the first derivation of, believe it's the mesoderm, that is associated with concentrating wastes, filtering them, and excreting them to the outside environment. So this larval protonephridia, we see the beating of these terminal cells and the flame cells, the beating of these flagella within it that change the concentration across the body wall, draw out metabolic byproducts and excrete them. So there's essentially an ultrafiltrate that is like urine, so that toxins, spent hormones, and urine is excreted to the outer environment. Now remember, up to this point, we've been dealing with either diploblasts that don't do this or simple enough, small enough organisms that simple diffusion has taken care of this. But now we're going to be talking about larger organisms that need these concentrating mechanisms to get rid of metabolic byproducts. So the beating of that flagellum, creating a current and a pressure differentiation that draws toxins in and filters them out. Now, one thing to consider is we've seen something that looked like this before, and briefly talked about it when we were talking about ctenophores, because they look like ciliary rosettes. And that's largely why, using some circular logic, why we think ciliary rosettes function like that. Now, if you think about the placement phylogenetically of ctenophores in the large context of the animal phylogeny, this will perhaps be another case where ctenophores have convergently arrived at a very similar looking solution to the rest of animal life. So we talked about that with regard to the through gut. We talked about that already with regard to the nervous system, and potentially here with excretion. You can imagine what in the same way that the ctenophore lecture had questions about, for you, that I posed about phylogeny and about the consequences of phylogenetic hypotheses. We'd already talked about the neural system and the through gut. So maybe another way to make questions for yourself to practice for that quiz is to do it with excretory organs like this. Same kind of question. So within the Spiralia, we've got annelids and mollusks, bryozoans, chaetognaths, rotifers, etc. And the molting animals, the arthropods, the nematodes, that's kind of the next group. This is a split here that is very much supported by molecular data, maybe more so than developmental data, as has been used to break up the protostomes and the deuterostomes. So the larval, the protonephridia, as an excretor. One of the other apomorphies of the Spiralia is the possession of chetae, or stiff bristles, that are composed of beta-chitin. And this beta-chitin is a specific kind of chitin in which the polysaccharide chains are arranged in a parallel fashion and not in antagonistically. And so we'll compare and contrast that when we get to Cedae within the arthropods and within some of the ecdysozoans in a few weeks. But this is an apomorphy for this particular group. Now, talking in big, large strokes here, I want to make sure that I jump over to the chat. And unless it's not refreshing, I don't see any questions. And so either everyone's kind of just dealing with their coffee on a Monday morning and a pleasant afterglow of having heard the beautiful voice of Attenborough this morning. I don't see any questions. We will keep going. We're going to change gears for a second here. And so when we change gears, this is in preparation for Wednesday's guest lecture about entomophagy. I wanted to have a chance to kind of prompt your brains and prime the pump so that when we receive our guest lecture on Wednesday that you have some questions and ready to kind of talk with the guest that's going to be talking to us on Wednesday. A reminder, what I said at the start, I'm going to put up in this case, kind of a post-lecture, 5 to 10 minutes to the YouTubes later on, where I'll talk about some of the last introductions to the platyhelminth worms. And that'll be a good idea to take a look at because it's tied into your labs this week. So, as the Python says, now for something completely different. Entomophagy. Phagy meaning to eat, entomo meaning insects, and so this is entomophagy. You can talk about entomophagous insects, parasitoids, for instance, that lay their eggs inside of developing stages of larval Lepidoptera or Coleoptera and maintain them as they eat them alive. But this is, of course, something that we've done, that our species does as well, and it forms part of the traditional diets for at least 2 billion of the people that you share the planet with. And as many as close to 2,000 different species of insects have been documented to have been intentionally used as a food source. And the screen grab on the bottom, this is the front of an FAO report that talks about the importance of entomophagy. The link I'll post in the chat later on if you're interested in it. But there is increasing interest in entomophagy for our species across the world due to an increasingly large population and energetic pressures associated with us generating protein as we traditionally do, at least in the West, with cattle and pigs and sheep. So you can see the map on the bottom is the recorded number of edible insect species by country. It's a strange map because it's broken up by nation and perhaps not by region. But I think one of the things to consider is that where insects are most diverse is where the greatest number of edible insects have been sourced by country. Now, let's talk a little bit more about where so that was by nation. This is maybe the graph now at the bottom is switched. So we're talking about edible number of insect species by order, by insect order, consumed worldwide. And you can see, so Coleoptera, these are your beetles. Lepidoptera, your butterflies and moths. Hymenoptera, these are ants and wasps and bees. Orthopterans, and so these large different orders. This roughly parallels, this frequency histogram of how many species by order are consumed worldwide is roughly corresponding to the diversity of insects worldwide. Take a look at the scale of the y-axis though, and realize that if for 30 insect species of beetle that have been consumed as, or documented by the FAO to be consumed as a food type worldwide, that's in relation to literally hundreds of thousands of species, maybe close to 400 or more thousand species. So a very small proportion we've explored. That might have to do with opportunity. Currently in the West, it certainly has to do with how we're trained to think about what's good food and what's bad food, but it may also have to do with size. Much of the diversity of insect life is very, very small, and so for our general body size, the human species, we're looking for larger things. So there'd be an interesting kind of an intersect for you to do an honors thesis to look at which species have been examined and their body size and their distribution, or their what proportion that represents, that are used as food, what proportion that represents of the total diversity. Now, why eat insects? Well, it's a protein source, and it can be as delicious as any other protein source that we eat. One of them, we can think about promoting it, or why it might be promoted, for 3 reasons. That and the FAO talks about their nutritious alternatives, so to mainstream staples such as chicken, pork, beef, and even fish. Rich in protein, good in fats, high in calcium, and they already form a diet, so that's a health reason. An environmental reason, that they can be promoted as a unit with fewer greenhouse gases than most livestock. That it's not necessarily a land-based, and so doesn't require us to transform, to degrade, or to fragment land. So, and ammonia production is reduced. They're very efficient at converting food into protein, and I think our guest on Wednesday is going to talk much more about this. And it's also insect harvesting, and I hope there's some questions maybe that you can pose for Ryan on Wednesday about these aspects. Insect harvesting is a low-tech, low-capital investment option that offers entry to even the poorest sections of society, such as women in the land list. This is speaking globally in terms as compared to protein generation from traditional stock animals. Now, many of you, some of you maybe, are sitting out there going, ooh, you're making me lose my breakfast talking about this as early as this in the morning. And we know that people have some people have a hostile response to the idea of eating insects. And in fact, there's some studies, and they're referenced there, and that'll appear in the chat later on, to how familiar or open you are to eating sushi as being a predictor of how acceptable your perspective is going to be to eating insects. So the frequency of sushi consumption, which is a food that was, when it was first introduced, was commonly met with disgust in the West, is a significant and substantial predictor of your acceptance to eating insects. So what I'd like to do today is talk amongst ourselves. The first one is going to be, we're going to speak via a poll, and you're going to tell me about your history eating insects and your perspective. So your first question in the poll, have you ever eaten insects? No, and I never will. No, and I'd like to try. Yes, and in fact, enthusiastic yes, they're in my smoothie this morning. Those are your choices. Begin the Jeopardy theme song. Oh, we're already at 89. Well, let's share the perspectives here. Awesome. I love this. Okay. So what we're seeing is about 48, slightly less than half of you have not eaten insects and are interested to try. A third of you have tried insects, and what I didn't put in there was yes and I hated it or yes and I loved it. 16% of you are not interested in doing this, and so we'll talk about that in the lab. And then 1% of you has insects in your smoothie, or at least acknowledges that if you're eating field-collected fruits and vegetables, there's a significant chance that you are eating it in your smoothie. Anyhow, I'm going to save that screen grab, and we're going to continue to the next. What I'd like to do is to use Zoom, the poll again. Share in the chat if you have eaten them, where and which kinds of insects. And maybe you've already started to do that. I had crickets from some random lady in a market in Cambodia, termites in Costa Rica, orthopterans and coleopterans here. So crickets, ants, wasps, all chocolate covered from a fine fruit grocery store in England. Scorpions as well. Cricket crackers, super good actually. This is amazing. The Butterfly Conservatory, yes. Which one is that maybe? Is that the Butterfly Conservatory that's sitting near us here in near Cambridge? We had enough crickets to kill a horse. I'm not sure if that's a unit of crickets, but that's amazing. And it was Cambridge, amazing. I've scrolled down and I got to a couple of other questions that I missed. I'll either circle back for these afterwards and try and answer them. But the one that I saw something saying that invertebrate isn't a taxon because it's just everything that isn't vertebrates. Is that true? You're right. Invertebrate is not a monophyletic taxon at all. And you can tell that by the position of deuterostomes in that Dunn phylogeny. Chocolate covered, amazing. So well done you. Now let's take, in the remaining seconds here, I'd like you to do one more thing. Jump into the breakout rooms, which I'm going to create here in a second. Talk about your experiences. Would you eat them? Would you not? And come up with some reasons, your own reasons perhaps, or share the reasons why you did or didn't. And then share them in that other sheet in the Google Doc. We'll spend about 4 or 5 minutes doing this. So to breakouts we go. Lollipops. A scorpion lollipop. That's a spicy lollipop. I'm seeing some of the answers for the discussion pop up in the Google Doc. And some of them are making me laugh. And I see some, I don't see any answers yet in the no, I would not eat insect column yet, but I see some cursors are hovering around there. So we'll give a couple more minutes. And I'll write them a message. Answering some chat questions. Okay, so I'm going to close the room and we're going to come back for a couple of seconds and I'm going to share some of your answers. Now, let's see. Hello, welcome back. I hope the feng shui of your breakout rooms and your discussion was enjoyable there. Can see everybody's coming back. Everybody's coming back. 30 more seconds. We got 2 more minutes. And after those 2 minutes, I'll tell you a little bit about what we're going to be doing on Wednesday. So I'm going to pull this across so you can see it. This is some of the, as you saw in that sheet, why would you eat insects? Yes, they taste good. Try things at least once, yes, for the environment, but I'm still a little worried. That's fine. Mixed with something else, at least for the first time. These are all prudent things. And I think really what you're coming up with is the same, it's a decision. If you don't eat animals, it's a recognition that these are animals. And so why would you change, if that's the principles that you're living your life by, why would you change because they don't have a backbone? So well done you. I think that's entirely fine. Now, in the last minute or so that we have together, I've just been in the chat, so I'm going to jump away from that for a second, and I'm going to jump ahead to the fact that We are going to be having Ryan Goldman from Entimo Farms, Ontario-based Entimo Farms. It's one of the largest producers of insect protein in the Western Hemisphere, and we're really lucky to have one of their founders, Ryan Golden, who's going to be talking about us on with us on Wednesday. So I'll post a bunch of these links that I've been using in today's discussion, and we'll take advantage, hopefully, of the time. He's going to give us, I think, a virtual tour of the farm. And big kudos to Sherry, and a thank you to Sherry for setting this up. I think this is going to be super cool. And it's the reason that in your kit that came with this course, there is, of course, that cricket powder. So think about some questions. Maybe explore the website, explore that FAO document if you've got your thoughts about how useful entomophagy may be for people. Think about some questions that you might want to ask, and we're going to be able to open up our mics and or share a chat and share questions with Ryan on Wednesday. So that is it for today. I'll have a 5 or 10 minute kind of post lecture where we'll keep talking about platyhelminthes to keep you thinking about this as you go to your labs later on in the week. And I'll encourage you to get outside and be good to your body, be good to your mind, read something that isn't social media or isn't associated with a course, and take care of yourself, take care of each other, and I will see you on Wednesday. Ciao for now.